Hello, everyone. I'm Dr. Duygu speaking again from Izmir, Turkey, for another topic for physiological chapter for academic performances and for board exams, as well as your clinical practice and as well as your scientific purposes. Let's talk about pearls of neurophysiology tonight. We have a big but also a brief information about why it is a brief information about neurophysiology, because it's the only a limited chapter for this. It's a brief information about it, but in fact, it's a huge constellation in the sky that we're talking about. So let's talk about neurophysiological pearls and ask your audience questions and get the responses to audience questions. So let's start talking about neurophysiological chapter. Can I take our slides, please? Okay. What we understand as we tell you neurophysiology, we know as neuron cells or neuromodulation is a new age for scientific research or for scientific purposes in order to understand what's happening in our brain or in our central nervous system, in order to expand what's happening inside our brains. Embryonic development process includes neural crest cells, which causes migration in order to form neurons, neural structures. As we know it, neural cells is the main elementary focus of neurophysiology. Let's talk about dorsal root ganglia is transforming to sensory system, and autonomic nervous system is also caused by neural crest migration. And in order to form neurons of mesenteric and submucosal plexus of GI tract, gastrointestinal tract, or alimentary tract is very important for neural structures inside our abdomen, of course. Everyone call it the abdomen or cerebrum inside our abdomen. How about cranial nerve ganglia is very important in order to expand the nature of neural structures and intercellular interactions. Let's you hear the neural plate in order to migrate to epidermis, for example, in order to shape neural tube, and neural crest cells migration here, dorsal and ventral aspect. You see here the dorsal aspect and the ventral aspect, of course, and the migration of neural crest in order to modulate the architecture of these cells, north dorsal root ganglia, autonomic nervous system, and submucosal and mesenteric plexus of GI tract, and cranial nerve ganglia. This is the main shape of neuron cells. You see here the dendrites and the cell body here, and the nucleus in the middle of the cell, and one myelin sheath in order to protect like an envelope, and like an axon and nodorenvia in the anti-axonal area, and one Schwann cell. And here the axon terminal is a very important area that I will expand in the nature and in the preceding slides. Let's talk about the neural cell structure and organization. And soma includes organelles like every cell body, in fact. It includes organelles like mitochondria, like Golgi apparatus, ER, blah, blah. How about dendrites? Dendrites includes afferent cytoplasmic connection area in order to shape the transformation of neural synaptic transmission. And it may recognize many of course, it's a very important area here. It's very important, of course, very important for or very high yield for board purposes and lab studies. One may recognize many dendrites across the neuron. One neuron has many dendrites, but for axons, one neuron has only one axon. Axon means it's afferent cytoplasmic connector area in order to transmit the signal to other neural structures like other neurons. This connection ends within synaptic terminals, as I showed you here. Here the axon terminal, and here is the synaptic terminal within the other neuron. And the key point here, as I told you before, each neuron has only one axon. Each neuron has only one axon. This is the key point, very high yield for exams. Here is the synaptic gap, is the mitochondrial organelle system inside the cell soma or cell body. This is the presynaptic axon terminals, and this is the synaptic vesicle here in order to shape the neurotransmitter, this regulation here. And you see the neurotransmitters, in the interneuronal area for neural transmission. And here is the ion channels, receptors, and postsynaptic dendrite in order to modulate the neural transmission. Neurotransmitters take part in the neurotransmission and mere part of signal transduction in order to fulfill the requirements for signal processing. Requirements for signal processing here for interneuronal modulation. How about neurotransmitters? You see, or you ask, or you read lots of neurotransmitters, but let's flesh out about them. These are peptide molecules in nature, biochemically, and produced in rough endoplasmic reticulum unit. Neurotransmitters are peptide molecules and produced in RER. Schwann cells. Schwann cells mean protecting and surrounding peripheral nerve axon with myelin structures. These are protectors of myelin structures. You see here, as I told you before, Schwann cells and surrounding peripheral nerve axon with myelin organization here. This is the protector envelope here. You can see that this electron microscopic slide. Let's talk about, by basic approach to neurotransmitters, the basic neurotransmitters. How about acetylcholine? Acetylcholine synthesized in the basal nucleus of Maynard in the central nervous system. As we know, clinically important, and for our board purposes and our clinical practice, it decreases in Alzheimer's disease, in Huntington's disease as well. But it rises, in contrast, it rises in Parkinson's disease. How about GABA? The Gaba is gamma aminobutyric acid, is very important for depressor in central nervous system. It's synthesized in nucleus accumbens area and decreases in anxiety disorders or general anxiety disorder, and decreases in Huntington disease as well. How about serotonin? Serotonin is very popular nowadays in order to research properties in depression, anxiety, and as well Parkinson's disease as well. It's synthesized in raphonucleus in central nervous system. As I told you, it decreases in anxiety, it decreases in depression, and it decreases in Parkinson's disease. How about norepinephrine? Norepinephrine is a kind of neurotransmitter, and it's synthesized in locus coeruleus, and it rises in anxiety, but unfortunately, it drops as levels in depression. Here is the serotonin pathway for serotonin biosynthesis. As we know, the precursor here is tryptophan, is an amino acid precursor. It's decarbonized here to 5-hydroxytryptophan by tryptophan 5-hydroxylase, and in order to build up 5-hydroxytryptamine, or other name is serotonin, by the enzymatic reaction catalyzed by aromatic L-amino acid decarboxylase. Here is the norepinephrine structures and biosynthesis. The precursor here is L-tyrosine amino acid, and L-tyrosine is hydroxylated by tyrosine hydroxylase, according to build-up L-dopa, and dopa decarboxylase catalyze another reaction in order to build up dopamine, then noradrenaline, and adrenaline. Here is the catalytic enzymes are very important for your board purposes. Here is the key point here is dopamine beta hydroxylase and phenylethylamine and methyltransferase enzymes are very important for your purposes in order to shape your answers in biochemistry, physiology, and pharmacology as well. How about dopamine? Dopamine synthesized in ventral tegmentum in central nervous system and drops in depression for levels. Dopamine levels drops in depression. But in contrast, it rises to peak concentration in schizophrenia and rises in Huntington's disease, and unfortunately it decreases in Parkinson. Drops in depression, rises to peak concentration is schizophrenia, rises in Huntington, and decreases in Parkinson's disease. How about Organelles Organization inside or within the neural structures? Smooth ERs in neurons work for protein folding. Proteins are folded in it and shaped into three-dimensional structures. And the function of Golgi apparatus in neuron is post-translational chemical processing and delivering the package to axons and in the axonal transportation in dendrites. And the mitochondria establishes concentration differences across some parts or inside outside the neuron by ATP processing. The key point here is ATP processing, making a concentration gradient inside and outside the neuron. How about the neuronal membrane? Another important structure here. It explains us excluding certain substances and chemicals in order to keep a shape of membrane and protect the internal structures or organelles here. And it has a shape, includes protein distribution and microstructures specific to neural cells, but it changes by the location, like dendrites and axon, any kind of neuronal area. How about cytoskeleton? Cytoskeleton includes three apparatus. One is microtubules, which is 20 nanometer diameter, and neurofilaments, which is 10 nanometer diameter, and microfilaments, which is the smallest one, is 5 nanometer diameter, which are very important for your board purposes and your clinical lab practice. These are the shape of microtubules and neurofilaments. Here, as you see here, the neurofilament structures here. And microfilament structures here, the shape here, like a processes here, and the centrosome structures in the central area of the neural structure of the soma. The axon starts with the axon hillock. It's very important. The hillock is like a structure, like a process anatomically, and it is called axon hillock. And another important feature is no rough endoplasmic reticulum in the axon. But sometimes ribosomes are the main characteristics of neural structures. And this main characteristic of axon area, no rough endoplasmic reticulum. But axonal membranes proteins are structurally different from the soma membrane. It includes that expands that it includes different proteins as a shape. And the end of the axon terminal is in order to form synapses, which is very important for neural transmission. The end of axon goes through the axon terminal. How about the axon terminal? As I told you, axon terminal, but what is axon terminal? It includes synaptic vesicles as 50 nanometer diameters, but there's no microtubule structure in it. But it's very enriched with mitochondria. That means it includes high energetic levels and ATP biosynthesis, and there are dense protein structures on synapses facing the other cell membrane. The other function is signal bringing and conveying the signal and the information in order to shape action potential. How about action potential? Is the main structure here for neural transmission and neural circuits. Actual action transmitters is very important for your intellectual abilities, for your mind capacity, and for your executive functions as well. The first here, the initial part is the triggering event, like a stimulus or like pain or fear or something like that. It causes sodium permeability and opening sodium permeable channels. Then sodium crosses across the membrane and causes depolarization. And at this moment, the plasmic surface of the membrane is less negative than the outer part of the membrane. And generated potential occurs. Here's a very important. And one generated potential occurs in order to shape the transmission and convey the information to other neural structure. The critical level for establishing one action potential here is called threshold. If the stimulus is under threshold, there's no action potential, unfortunately, and there's no neural purpose, unfortunately, as well. So everything here, there was a donut shape structure diagram was here, but I couldn't expand it here. I'm sorry for that, but testing, falling in love, seeing something, singing a song, playing the piano is everything, and every executive function is connected with action potential and one threshold as well. Here you see another routines of neural interaction is on some routine neural studies, action potentials are created artificially via microelectrodes transferred in order to pursue an artificial neural functioning. These are attached to the neural cell's body external cell membrane in order to create an action potential for some research studies and in order to expand the conclusions. And after generating an action potential, there's approximately 1 microsecond time with a pause period, which is very important, with a pause period in order to create a new action potential, which is a pause period. So you won't able to create any new action potential. That is called absolute refractory period. It is absolutely a refractory period. So you won't able to build up any new action potential in this period. It's like a resting period. It's like a fiesta in Spain. So here is the source, siesta in Spain. Here's a Van Gogh Starry Night picture. As you know, this drawing is one of the most famous ones of Van Gogh. But art is very connected with action potentials as well. If your brain structures, your neural structures doesn't work properly in an accurate way, so if your neural modulation don't function well for one stimulus, for other triggers, so if you don't have any neural structures and if you don't have any action potentials, you don't draw any pictures like that, very world famous and very important pictures for all of the world. So another art, as you know it, is sculptures, for example, performed by Rodin. August Rodin's Thinking Man, like others, very famous structures from antique age to our nowadays for the century. So if you don't have any action potentials or executive functions created by action potentials, there will be no art. Another thing is playing the piano, as I told you before, and running, like sportive functions, like for example, Olympic games or anything else. You won't be able to work, or you won't be able to run or swim. You won't exclude anything else without neural stimulus, neural triggering, neural thresholds, and action potentials. And I appreciate your interest, your precious interest for everything, and your patience. And I will happy to answer your questions. You can send us by email, and you can write to our academic social media. So you can be in contact with our distributors, with our editors. You can ask any questions. I will be happy to respond any question coming from audience. Thank you, and have a nice evening. See you in the next information I'll told you. And in the nowadays and the other lessons, I'll give you more information about physiological and pharmacological approach for your board purposes and for your clinical practice as well.